I'm going to review transcription and then record translation for you. So transcription, the message is transcribed. And basically, what holds the original message? DNA. DNA is used as a template to decode the message by creating what? RNA, okay? How does this occur? What happens first? DNA unzips, okay? Now, if mRNA's strand consists of A, T, C, G, A, oh my God, it's not mirroring. U, A, G, C, U. Okay? Right here. All right? Where is all this happening? The DNA unzipping, the making of RNA? In the nucleus. Because we know that DNA never needs a nucleus. So this happens. Now, once this happens, RNA gets modified, and by that it means whatever errors occur or whatever needs to be fixed happens, and RNA leaves the nucleus. Never. What is RNA leaving the nucleus for? To go find a ribosome to complete the process of protein production. Okay? Question. All right, so we move on. So now we do translation. The code is interpreted. The code that originally came from DNA made RNA to signal specific what? tRNA is attracted to RNA, but it signals to bring what? Amino acids, okay? So mRNA, as you mentioned before, moves into the cytoplasm to attach itself to a ribosome. Together, they're going to finish the production of protein. How many letters in RNA 3 does it take to read the code? And it's 3. What do we call these 3 letters on mRNA? Codons. They're codes to be interpreted, and each codon specifies a specific amino acid, okay? Who brings in the amino acids? tRNA. tRNA holds nucleotides, okay, or is made up of nucleotides, and the N bases are what in comparison to RNA? Opposite or corresponding, attracting each other, yes. And they're called anticodons. Good. So we're moving on to the next slide. The anticodon is on tRNA and it's attached to the amino acid, okay? Anticodon is corresponding N bases. So if the codon is A, U, G, G, C, what's the anticodon? U, A, C, C, G. Good job, okay? Now remember that these links, if you want to watch them again, open the notes in iBooks and copy and paste the links, okay? That's the only way you can get the PDFs to become active. Now, we are going to actually, I'll record this and we'll call for specific amino acids so you can watch it happen, because this is definitely going to be on the test, okay, like we did in class. So let me make this bigger if I can, okay? All right, so what's this strand? mRNA. How do we know that? Because it's single-stranded. And look what this word says. It's part of translation. There's no more DNA because we're already in the cytoplasm. And what's this strand looking for? A ribosome first, okay? So it's going to look for a ribosome, and then it's going to start the process of protein production. And I believe it starts here, AUG. Let's see. If it does start in AUG, what amino acid is it going to call for? Go to your charts and check. And I'm going to go over there. Who can find AUG? Yes. MET, right here. Okay? Let's see if that's real. Who's bringing MET? tRNA. Let's see if that's real. What's on tRNA? What's on tRNA? The amino acid and the anticodon. What's going to be my anticodon? UAC. All right, let's see. I hope I'm in the right place. I am. Thank God. UAC, tRNA, and here's the amino acid you said it would call for. All right, next. AAG. What's coming? Let's go here. What? No, AAG. Is that what the code is? Because I left the page. Let's see. AAG is calling for LY. Where is it on the chart, guys, so I can show it in the video?
Right there. The AA. Third of A.
Third row here.
Yeah.
Here it is, okay? All right, who's bringing that? tRNA. And what's tRNA's anticodon? UUC, okay? Let's watch it. I gotta press play. There's UUC and there's lysine. All right, and then again, it's just gonna go on, and these amino acids are gonna combine to form proteins. Questions? There's 64 codes, okay, because you have 4 nitrogenous base and 3 letters at a time makes 64, but there's only 20 amino acids. So you see here that, let's say, this amino acid is called by 4 different codes, okay, or codons, I should say. All right, so moving on. Mutations. This is not as perfect as I made it seem. Things happen. When DNA makes RNA, there could be an error, and we saw this when we studied the cell cycle, okay? And these are some examples of errors that can occur. One is called, and they all have to do kind of with letters, point mutation. One letter of the code is incorrect, and sometimes it has no effect at all. So here is the code ATG, and it makes UAC. Is that correct?
Yes.
Yes, okay. But here we've got ATA makes UAU. The error's right here. Something happened to this G, and instead it became an A. And over here it became a C. Hold on, I need to see this. When you have that mutation, this particular code can say stop making protein, when in general it shouldn't have. So the making of protein would be affected. Does that make sense? This, on the other hand, has a larger effect. When you have a shift in the whole frame. And I use this sentence here, the cat ate the hat, and look what happens when you shift a letter to that whole sentence. It warps the whole message. So if you shift the frame, you're shifting the whole message. Everything is interpreted incorrectly. It's not just one part of a code, it's the whole code. So what's going to happen, either an N base is going to be added or an N base is going to be removed. And I know this question. How? Because these are chemical reactions. When nucleotides are combining, sometimes they combine incorrectly, yeah? When DNA is splitting and making a copy, sometimes errors occur. Okay, questions? So in this case, N base was deleted right here. So they shifted the whole entire code. So you know, let's say this was 8. I mean, I tried to make words because I wanted you to see how warped the word becomes, yeah? But if this was AUC, and that calls for a specific amino acid, and then now it's AUG, and everything after every 3 codes is wrong. So it causes serious effects. All right, other mutations happen during meiosis, and we didn't really talk too much about this because normally I do projects, but you can get an extra chromosome, right? Have you heard of trisomy 21, which is Down syndrome? Mm-hmm. So basically the 21st pair of chromosomes gets an extra chromosome during meiosis. So when they line up together, you get an extra one. And so that's how you end up with Down syndrome. And I think that we are done. And that is